The last part of the membrane structure material that we'll talk about before we start talking about membrane transport is the arrangement and the different types of proteins that we find in the membrane. The most important thing to remember about membrane proteins is this: they are very abundant. Remember, there's just as much protein in a membrane as there are phospholipids. Now, you learned about the different categories of membrane protein in BIO312 molecules and cells. And just like we did with the phospholipids, we're going to add some more detail today into your understanding of membrane proteins. If you recall from BIO312, we talked about two basic types of membrane protein. We talked about the kind of membrane proteins that go across the membrane. We called those transmembrane proteins. And we talked about the kind of proteins that stay on one side or the other. And we called those peripheral proteins. We're going to broaden our discussion today with a few more categories. The first category of membrane protein we're going to call an integral membrane protein. The transmembrane example is one of those types, but not the only type. So an integral membrane protein is any membrane protein that goes into the membrane at least to some degree. In other words, it penetrates through the head region and into the tail region. That's an integral membrane protein. An integral monotopic protein is one that penetrates the head region into the tail region, but only across one monolayer. That's what we're going to refer to as an integral monotopic membrane protein. In comparison, a transmembrane protein is one that crosses the whole bilayer. So it penetrates through the heads on one side, crosses through the tail section, and penetrates the heads on the other side. That's a true transmembrane protein. Now we're going to add a little more detail here with these terms, because some membrane proteins will cross the membrane, again, the entire bilayer, one time. In other words, you'll see one passage across the bilayer with this protein. That's called a single pass membrane protein. But remember, proteins are amazingly complex in their size and shape, and some transmembrane proteins will actually turn around and cross the membrane more than once. Those are called multipass transmembrane proteins. In other words, the protein penetrates all the way across the membrane once, folds over 180 degrees, and then penetrates the membrane a second time, and sometimes a third or a fourth or a sixth or a 10th time. Those are multi-pass transmembrane proteins because they are crossing the bilayer membrane multiple times. Now, a peripheral protein is a protein that does not penetrate into the tail section. It stays in the area of the heads. Now, some peripheral proteins will sit in and among the heads of the outer monolayer, and some will sit in and among the heads of the inner monolayer. Those are peripheral proteins. There's one other category, and that's what are called lipid-anchored protein. A lipid-anchored protein is going to primarily sit around the head region on one side of the membrane or the other, but importantly, it will be bound to the lipids in that portion of the bilayer. Remember we talked about lipid rafts, and we said that there are regions of membrane where the proteins stay. They stay put. And the way that's achieved is by anchoring them in place, anchoring them to lipids in that area, to phospholipids in that area. Let's take a look at a diagram of some of this variety of membrane protein. In this image, the artist has drawn the lipid bilayer in purple. So here's a monolayer with heads and tails, and here's a monolayer with heads and tails. And this is the bilayer. We're going to imagine that this is the outside of the cell, this is the inside of the cell. Now let's take a look at some of these proteins, which are all depicted as these squiggly lines. I like this drawing because it reminds us that proteins begin as a chain of amino acids. A polypeptide is simply a long chain of amino acids that have been covalently bound to each other with peptide bonds. That polypeptide is then going to be folded up into alpha helices and beta sheets, and then those will interact with each other through hydrogen bonding and van der Waals interactions, and we will get a three-dimensional protein. Remember, every protein has its own unique three-dimensional shape, and that means every protein has its own unique function. So take a look at this brown protein right here. The way this protein has folded up, part of it sits out by the head region of the outer monolayer, part of it sits right in the tail region, and part of it sits out in the tail region of the inner monolayer. This is a transmembrane protein, and it is a single pass protein. Now, we can tell something about the chemistry of the amino acids in this protein just by the fact that it can cross the entire bilayer. We know, for example, that the amino acids that are right in this region must have R groups that are hydrophilic, that are polar in their chemistry, because this region of the protein is literally exposed to a watery environment, polar, and to the polar heads of the membrane. So just by the fact that it can sit here like this, we know a little bit about its polar nature. Now compare that to this part of the protein in here. In order for this region of the protein to be able to sit right here amongst all these tails, the amino acids in this region must be non-polar, hydrophobic. And then at this end, we must be back to polar chemistry. So remember, proteins, just like phospholipids, are amphipathic. They can have both chemistries, both polar and non-polar chemistries, in the same molecule. Transmembrane single pass. Over here, here's another transmembrane single pass. Here's a transmembrane multipass. Now you have to follow this green string to really see the multipass nature. Again, part of it over here all begins outside, on the outside of the cell, in the watery polar environment, and then it crosses the membrane right here. When it gets over to the other side, it folds and then comes back and crosses a second time, and then it folds over and comes across a third time, and so on and so on. This is a multipass protein. Again, we know something about the amino acids, the order of the amino acids in this protein. we know that any amino acids that are sitting here in this part of the protein or here must be polar, and that these amino acids must be nonpolar. This protein right here, depicted in this green color, this is a peripheral protein. It's not interacting directly with the heads, but instead with the polar region of another protein. Sometimes that happens. Sometimes peripheral proteins sit all by themselves right among the heads of the membrane. Over here we can see one of these lipid anchored proteins. Again, the bulk of it looks like a peripheral protein, but it is anchored in to the tail region. Here's another one depicted as an anchored protein. This happens to represent a glycolipid. The little squares are supposed to indicate there's carbohydrate attached here, but you can see that it's anchored in to the membrane. Here's another peripheral protein that again happens to be interacting with the polar regions of this transmembrane single pass protein. We see all different types of shapes and sizes of proteins embedded in membranes and associated with membranes. Now we're ready to move forward and talk about membrane transport. The variety of structural features that are in a membrane, whether it's a plasma membrane or a membrane around an organelle, the variety of different features are going to determine how permeable that membrane is and whether or not individual molecules can get across. Membranes are selectively permeable. Depending on the membrane, depending on whether it's a plasma membrane or an organelle membrane, and depending on the type of cell, permeability is going to vary. If you recall from BIO312, we said that certain molecules have the ability to cross a membrane simply by passing through the phospholipid molecules. Other molecules are going to require some assistance, and that assistance comes from the membrane proteins that we've been talking about. So we're going to be talking about 3 modes of passage across a membrane as we discuss membrane transport. We're going to talk about simple diffusion, we're going to talk about facilitated diffusion, and we're going to talk about active transport. And just as we've said for the other parts of today's lecture, we're going to review what you already know about these modes of passage, and we're going to add a little bit more detail today. So take a look at the drawing at the bottom of the slide for a minute. You can see a section of membrane bilayer depicted here. This is representative of the extracellular environment, the outside of the cell, and this is representative of the inside of the cell. You can see the heads drawn in this beige color on both sides of the bilayer, and the tails drawn in light yellow. You can see a bright yellow depiction of cholesterol distributed through the membrane, and you can see purple proteins. You can also see some carbohydrate on the outer monolayer of this membrane. What they're showing us in this slide are a variety of transmembrane proteins that are involved in this kind of facilitated passage across a membrane. So yes, some molecules are going to cross the membrane by literally passing between the phospholipids, but other molecules are going to need the assistance of these membrane proteins. Let's start by talking about simple diffusion, the type of membrane passage that involves a molecule crossing a membrane in between the phospholipids. Simple diffusion is a spontaneous process. That means it does not require any energy in order to occur. The only thing that drives simple diffusion is a concentration gradient. The direction of transport and the speed of transport are going to be determined by this concentration gradient. And if you recall, all a concentration gradient is a difference in the concentration of some kind of solute between two different regions across a membrane. So we're comparing perhaps outside the cell to inside the cell. And if there is a difference in concentration of some dissolved particle, some solute, then we have a concentration gradient. And at least the possibility exists for simple diffusion. The direction and the speed will be determined by that concentration gradient. But whether or not simple diffusion can occur is going to depend upon the chemistry of the particle trying to get across. Simple diffusion is very effective for small molecules that are non-polar and do not have a charge on them. By small, we mean less than 100 molecular weight. So, good examples of molecules that can get across a membrane using simple diffusion are things like oxygen gas and carbon dioxide gas. Remember, those gases are able to use simple diffusion because of their chemistry. They're both small molecules with a low molecular weight. They are non-polar and they don't have a charge. So they are capable of simply slipping across the membrane between the phospholipids, from the outside of the cell to the inside of the cell, and from the inside of the cell to the outside of the cell. As long as there is a gradient in place, they can cross through simple diffusion. Simple diffusion is always going to move the solute, the dissolved particle, down the concentration gradient. And remember what that language means. It means from the side with the high concentration of particle, across the membrane, to the side with the low concentration of particle. From high to low until equilibrium is reached. Remember, nature abhors a gradient. Nature would like to get rid of all gradients and get things to equilibrium on both sides of the membrane. Simple diffusion is spontaneous. You don't need to add energy to make it happen. The gradient is driving it, and it will continue to happen until equilibrium is reached on both sides of the membrane. Let's take a look at this diagram. This diagram is representing simple diffusion across a membrane, but we read it from left to right because in the diagram, time is passing from left to right. So we're going to look at this membrane, this bilayer here, and this dissolved particle, this solute that's shown as a blue circle. This is the outside, the extracellular fluid. This is the inside. This is where the cytoplasm is in the cell. So we're starting off with a whole lot of blue particle out here. and none, no blue particle inside the cell. That's a pretty extreme gradient. Higher concentration here, lower concentration here. We're told that this blue particle is small, it doesn't have a charge, and it doesn't say it, but we can also assume that it's non-polar. It's going to meet all three of our requirements for simple diffusion, and there's a gradient. Remember, the size and the direction of the gradient is going to determine the direction of simple diffusion. The blue molecules are going to move from the side with a high concentration across the membrane to the side with the low concentration. All right, now let's watch time pass. If we go to this middle panel here, we can see simple diffusion occurring. The blue molecule is passing across the membrane just right between the individual phospholipids, from the side with the high towards the side with the low. And if we check back as more time has passed by, simple diffusion has finally stopped. The only reason it stopped is because the gradient is now gone. We have equal concentrations on both sides of the membrane. We are at equilibrium for this blue particle. Simple diffusion has done its job, and it now stops. So in order for simple diffusion to occur, there has to be a gradient in place. And in addition to the gradient, the molecule, what we call the solute, because it's dissolved in water, the molecule has to have these three chemical features. It has to be small, and we define that as having a molecular weight less than 100. It has to be relatively nonpolar, and it has to be uncharged. It has to be neutral. Now let's talk a little bit more about this chemistry and why it matters. I think the size piece of it is pretty obvious. A big, bulky molecule is going to have trouble pushing in between those phospholipids. So the smaller, the better. When it comes to polarity, remember that when we call a molecule polar or nonpolar, we're referring to the chemistry of the covalent bonds within the molecule. We're referring to how electronegative the atoms are within that covalent bond. If there's a big difference in electronegativity between the two atoms that are bound together, we're going to see partial charges forming, and that particular covalent bond will be polar. When we have two atoms that have the same electronegativity, and they are sharing a covalent bond, that will be a nonpolar covalent bond because it has no partial charges. Now, polarity and non-polarity is actually on a spectrum. Depending on the molecule and depending on how many of its covalent bonds are polar and how many are non-polar, that will determine the overall polarity of the molecule. So it's a spectrum from very polar to very non-polar. Now you can actually measure polarity by determining what's called the partition coefficient of a molecule. And all that is a comparison of its solubility in oil in a non-polar solvent divided by solubility in water or a polar solvent. The more non-polar a molecule is, the higher this top number will be, and the lower the bottom number will be. Remember, non-polar things dissolve in non-polar solvents like oil. So the more non-polar the molecule is, the bigger this number will be, and the smaller this number will be, so the larger the partition coefficient. When you have a polar molecule, this will be the bigger number because water is polar too. And that will give you a smaller partition coefficient. So nonpolar things have higher and higher partition coefficients. Polar things have lower and lower partition coefficient values. Small molecules, relatively nonpolar, so relatively high partition coefficient, and no charge. Now, why is it important that these molecules have no charge associated with them? Well, it relates to water. All living cells are in a watery environment. And if you take a charged molecule and you put it into a watery environment, the polar water molecules are going to interact with the charged solute. Remember, water, because it's polar, contains partial charges, partially negative oxygen and partially positive hydrogen. Depending on the charge on the solute, it will either be attracted to that oxygen, partially negative, or it will be attracted to the hydrogens, partially positive. The result is the same, though. If you have a charged solute, it will hydrogen bond with water, and you end up with what we call a hydrated molecule, a molecule that is surrounded by water with hydrogen bonding, and therefore a large, bulky thing. So charge is about whether or not the solute is associated with water molecules. Any charged particle is going to have trouble getting across a membrane because it will become hydrated. On this slide, we're looking at this phenomenon of simple diffusion, which is that the concentration gradient is going to determine the direction of flow and the speed of flow. On this graph, I want you to first look at this red line, which depicts the kinetics of simple diffusion. Notice that it's a straight line. This is a linear function. On the x-axis, we're looking at the size of the concentration gradient. And on the y-axis, we're looking at how fast simple diffusion is occurring. So, as I get a larger and larger x value, I get a larger and larger y value. That's what you're looking at here. As x gets bigger and bigger, y is also getting bigger and bigger. See that? I have a linear function. As concentration gradient increases, the speed of simple diffusion increases. And that relationship just keeps going. Okay, there's no end to it. There's no plateau. Now, there is a very special case of simple diffusion that involves water. And that special form of membrane transport gets a name. We call it osmosis. So osmosis describes the movement of water across a semi-permeable membrane. The reason that we separate out the behavior of water is because we can't talk about water in simple diffusion terms, because we can't talk about a concentration gradient for water. We can only talk about a concentration gradient for the solute, for the dissolved particle. Osmosis occurs in response to the dissolved solute, the particle. Osmosis is the movement of water across a membrane in response to the distribution of particles across that membrane. Just as we said that simple diffusion will proceed until there is an equilibrium across the membrane, osmosis will also proceed until equilibrium gets reached. Take a look at the diagram on this slide, which represents osmosis occurring in a beaker. So what you're looking at on the left-hand side is a beaker that has a membrane stretched across the center of it. You can see that the beaker has been cut in half, so we can peek inside. Now there's a green solute in here. There's a green particle dissolved in water. And I think you can see that there are fewer green particles on this side and more green particles on this side. So there's a concentration gradient in place across this membrane. Now if this green particle was small and relatively nonpolar and it didn't have a charge on it, we would expect simple diffusion to occur. We would expect this particle to move from the side with the high concentration across the membrane to the side with the low concentration. But now we're going to imagine a situation where this particle can't use simple diffusion. Perhaps it's too big. Perhaps it's too polar. Perhaps it has a charge associated with it. Now we have a problem because now the gradient cannot resolve. The gradient cannot go away by this green particle crossing the membrane. Remember, nature doesn't like a gradient. Nature likes equilibrium. So what's going to happen? The answer is osmosis is going to happen. Osmosis occurs in response to a particle gradient that cannot resolve by itself, cannot resolve through simple diffusion. Now, what makes osmosis strange is that the water will move in the opposite direction than what we would predict for particle movement. We would predict that particles would go from the side with the high concentration toward the side with the low concentration. Water is going to do exactly the opposite. Water is going to move from the side where there's a low concentration of particles towards the side where there's a high concentration of particles. Remember, nature wants equilibrium to occur on both sides of this membrane. An equilibrium of concentration, number of particles per volume of water. If the particles can't move, the water will, and it will move towards the particles that have a higher concentration so that they will become more dilute. Take a look at the beaker after some time has passed. The water moved from the side with a lower concentration of particles toward the side with the higher concentration of particles in order to dilute out that high concentration. I think you can see that they've drawn the green particles now so that you can see the concentration is the same on both sides. Even though you still have more particles over here, because there's now more water over here, the number of particles per volume of water is now the same on both sides. That is why osmosis occurs, in order to equalize concentration on both sides of the membrane. Now, living cells are not beakers. Living cells are in fact closed systems. They are surrounded by a membrane. And we have to remember that when osmosis occurs, it can actually be kind of devastating for a living cell. Organisms are built in a way that helps prevent too much of this osmosis from going on because it could be detrimental to the health of a cell. For example, if too much water were to leave a cell through osmosis, that cell would sort of shrink down upon itself. And if that shrinkage went too far, the cell would actually die. If too much water were to move through osmosis into a cell, the opposite thing would happen. That cell would start to swell. And if a cell swells up too much, it's going to rupture and the cell will die. This brings us to a concept in cell biology called tonicity. Tonicity is a measure of particle concentration, specifically looking at the outside of the cell relative to the inside of the cell. So tonicity looks at a difference in particle concentration, again, specifically from the outside to the inside. And we can describe 3 situations of tonicity. What's called isotonic conditions, where the outside particle concentration is the same as the inside particle concentration. We can describe hypertonicity, where the outside concentration is higher than the inside. And we can describe hypotonicity, where the outside concentration is lower than the inside concentration. So if you look at these words, if you translate them, the tonic part of the term means tone. Iso means same. So same tone means same concentration on both sides of the membrane. Hyper means more or increased. So hypertone means that there's more particles on the outside compared to the inside. Hypo means less or decreased. So less tone on the outside compared to the inside. Fewer particles on the outside compared to the inside. These 3 states of tonicity really matter when you're talking about a cell because, again, they can lead to osmosis. And while cells can tolerate a small amount of osmosis, they cannot tolerate extremes of osmosis because that can damage and kill a cell. So if we start on the left-hand side, we can see an isotonic solution